Hello, my name is Christina Knight, and today I will be presenting on autism spectrum disorders, or ASD. First, I will provide a conceptual review of ASD, which on a very general level is a group of developmental conditions that often begin to manifest in childhood and cause long-term damage to individuals. ASD is diagnosed based on a triad of behaviors: impaired social interaction, lack of communication abilities, and repetitive and inhibited activities. Cognitively, ASD reflects deficits in social cognition, such as theory of mind, which is the ability to understand other people and what they're thinking, and emotion processing, and primary deficits in non-social / domain general processing, such as executive dysfunction, which is the cognitive control of behavior. In 1943, Leo Kanner first emphasized these traits by describing autistic aloneness and insistence on sameness. These traits occur together in autistic people, but they are not obviously connected and are actually quite different. Former ASD behavioral, cognitive, and genetic research rests on the assumption that these three attributes should be explained together. However, new findings, and specifically our presentation, suggests that studying this triad of traits separately provides more valuable insight into future ASD research. In other words, ASD may not have one singular cause. In this portion of the presentation, I will delve into findings and experimental techniques used for behavioral, genetic, and cognitive research on ASD, focusing on findings that corroborate the assumption that no single cause ties these three traits together, focusing specifically on a heterogeneity twin study. Then I will briefly introduce my partner sections. First, I will discuss the behavioral research. Even though the co-occurrence of these three traits in an individual indicates ASD, individuals in the general population also often exhibit one or more of these traits. Studies that have measured ASD characteristics in the general population and those clinically diagnosed with ASD have found a smooth continuum from the regular population to clinically diagnosed individuals. This is important because this means there's no large spike or hump at the extreme of the spectrum. However, the general population usually exhibits just one of these traits. For instance, 59% of individuals who exhibit social impairments only exhibit social impairments. But if an individual has one of these traits, they have a higher chance of exhibiting the other ones. So 32% of those with social difficulties also have communication difficulties. There are different developmental paths of these traits also. So the repetitive rigid behavior occurs later on in life, whereas social and communication impairments manifest early on in childhood. These behavioral traits are also shown genetically. I will touch on one study cited in a paper about genetic heterogeneity. My other partner, Lenny, will also speak about the hereditary nature of ASD in greater depth. The paper I looked at is called Genetic Heterogeneity Between the Three Components of the Autism Spectrum, a twin study. In this paper, Ronald et al. Hypothesized that the causes of the three ASD characteristics would not be fully correlated genetically. They gathered parents of 3,400 pairs of monozygotic and dizygotic, which just means identical and non-identical 8-year-old twins from the twins' early development study, who had completed the childhood Asperger's syndrome test. This exam screened general population children for ASD symptoms. Then, Ronald et al. Reported genetic model fitting of categorical and continuous data. They found that extreme ASD traits had high hereditability and little correlated environmental influence. Further, all three of these characteristics had hereditary causes, but little overlap existed between these hereditary causes. Surprisingly, even social impairment and communication difficulties, which seem the same at first glance in everyday life, had low relativity. So they had a correlation R value of 0.2 to 0.4. Also, more than half the genes necessary for social disabilities were independent from those that contributed to the other two traits. This link between social impairment and communication difficulties have the same strength as the connection between communication difficulties and rigid behavior, which had an R value of 0.3. So there's a low correlation between all three of these ASD traits, both in people clinically diagnosed with ASD, but also the general population. Therefore, each of these traits had almost distinct genetic influences. Ronald et al. Concluded that the triad of ASD characteristics have heterogeneity in their genetic causes. This means that autism is not specifically hereditary, but there exists a broader autism phenotype, which is a manifestation of all or part of these traits in one's genetic code. The findings of Ronald et al. Are important because they are also reflected neurologically. These neurological findings are often reflected through neuroimaging studies found through MRIs and EEGs. MRIs are being used more and more often to study and detect autism. However, there are very distinct regions of the brain that are linked to autism. There's no one specific region of the brain that is directly connected to autism. So social cognition relies on the medial frontal cortex, which is in this region, this blue region in the front, the temporal parietal junction, which is where the parietal lobe meets the temporal lobe right here, where the yellow and the, I'm not sure if you can see my mouse, but where the yellow and the green meet, and the temporal pole, which is the interior end of the temporal lobe, down here at the end of the green section. And these are all very different from the regions of the brain that are linked to rigid and repetitive behavior, which is found as a caudate or abnormality in the basal ganglia, which is the center of movement. The lack of attention to speech, i.e. communication impairments, are associated with abnormalities in the superior temporal sulcus, which is this region that I've labeled right here, this fold, the sulcus in the green area on the map. This region was found through EEG. So these neurological studies also found that there's no one specific region of the brain that's linked directly to ASD. Instead, each of these specific traits reflect abnormalities in different regions of the brain. Now, I will introduce the areas of ASD that my partners will expand on by addressing the gap. First, Lenny will also provide a study that focuses on heredity. ASD is one of the most heritable neurodevelopmental disorders that creates impairments in social cognition. Lenny's approach to reviewing the hereditariness of ASD looks at brain morphometry and compares brain structures between ASD and non-ASD groups. Research using MRI scans can help increase our understanding of whether the cortical dysfunction of the brain comes more from developmental disruptions to the brain's social regions or more from improper interactions in one's environment. Next, Deba will review the sensory symptoms that occur as consequences of ASD. These symptoms are often overlooked. For instance, the diagnostic criteria for autism was only very recently revised to include sensory processing as a key domain of the autistic experience, though sensory symptoms have been observed in early reports of ASD conditions. As such, there's not enough literature informing the processes underlying the sensory side of autism. This is important because we actually stand to gain much insight into circuit level alterations in the brain from studying this gap in knowledge. And this may help to create translatable biomarkers of autism. Thank you so much, and I hope you enjoy learning more about these distinct traits that are relevant for ASD research. Thank you.
All right, so now I'm going to begin my section of the presentation by discussing ASD and its heredity. In this section of the presentation, The goal is to understand what additional research may be more helpful in closing the knowledge gap in our understanding of the genetics of ASD and its effect on the brain structure. In particular, how current studies are finding correlations in the brain structure and the genetics of parents with children with ASD versus parents with children without ASD. Some of the terms I'll be using to break down throughout the course of the presentation are theory of mind, brain morphometry, endophenotype, AQ and SQ scores, and MASD. And as I go through the presentation, I'll cover the important concepts and findings. So to begin with, autism spectrum disorder, known as ASD, is a neuropsychiatric developmental disorder that produces abnormalities in social behaviors. Those with ASD are most characterized by social avoidance and disinterest in social interactions. Some of the common symptomatic behaviors include repetitiveness, deficits in communication, and language development delay or language development absence. ASD affects approximately 1 in 68 children in the United States. And because it's one of the most common psychiatric disorders, this means that genetics play an important role in its causation. Some of the earliest evidence of ASD comes from infants that express impairments to their selective attention. And ultimately, the cumulative impairment underlying the social behavioral challenges with children with ASD comes from their theory of mind, which is the ability to understand others' thoughts and feelings as well as their own. So if one's ability to take inputs from their environment and respond appropriately is impaired, then it is believed that impairments to social cognition in children with ASD might be brought on by some dysfunctions in the cortical regions of the brain. So the image to your right pinpoints some of the functional brain regions that are related to social behavior and could thus be impaired in those with ASD, such as the anterior insula or the interparietal sulcus. It is believed that cortical dysfunction is to blame for the poor development of the cortex, which houses the social regions of the brain. However, where the research begins to run dry is in understanding the cortical and subcortical brain regions in prenatal and early postnatal processes, so before and after birth. Does disruption of the brain development lead to social abnormalities, or does improper interaction with one's environment lead to undeveloped social related brain regions? So knowing this cause and effect relationship has been difficult to parse out. There have been some studies on infants that show differences in the brain morphometry between those with ASD and those without. And by brain morphometry, I mean the study of the structural measures of the brain, such as its volume and shape. So to give a quick example, a longitudinal MRI study looked at the morphology of the corpus callosum in infants who were both at high risk and low risk for ASD. And the study found that those at high risk of ASD had a larger and thicker corpus callosum area and were later diagnosed with ASD beginning as early as 6 months of age. This is just one example of a study looking at children with ASD, but overall, few studies have looked at parents of children with ASD. One kind of research experiment in particular decided to take a more closer look at parents. So a study by Ann et al. Recruited 46 healthy mothers of children with ASD. This group is referred to as MASD. And they also recruited 48 healthy mothers of normal developing children, so children without ASD. And this was their control group. Researchers obtained structural MRI scans and performed voxel-based morphometry, VBM. The purpose of their research was to study the parents of individuals with ASD and better understand the different endophenotypes of ASD. An endophenotype is a heritable trait which is used to describe the connection between a disease's symptoms and the genes associated with the disease. So the researchers had two objectives here. The first was to assess whether the MASD group showed atypical brain structure patterns compared to the control group. And the second objective was to determine whether brain volume abnormalities in MASD were correlated with AQ and SQ scores. AQ is known as the Autism Spectrum Quotient, and it's a diagnostic questionnaire that measures autistic spectrum traits. And SQ is the systemizing quotient, which measures general autistic traits and the drive to systemize. In their results, researchers found no significant difference between the AQ and SQ scores between the two groups, but the VBM analysis revealed that gray matter volumes in several brain regions were smaller in the MASD group relative to the control group. Specifically, the smaller gray matter volumes in the MASD group came from the right side of their middle temporal gyrus, the right cerebellum, and the parahippocampal gyrus. From this study, researchers suspect that systemizing autistic traits can be correlated with smaller volumes in the social parts of the brain, in the middle temporal gyrus or in the temporal parietal junction, and that atypical gray matter volume from these social parts of the brain may represent potential neuroendophenotypes of ASD. So to give some visual to what I just described, the image in the top left of your screen shows which parts of the brain had decreased gray matter, and those parts are marked in red. The second image in the upper right shows you the first cluster where decreased gray matter was found. So this is in the middle temporal gyrus of the brain, and that is marked in the yellow. And the third image at the bottom just shows you how significant the gray matter decrease was in the MASD group compared to the control group. So the final part to close out this section of the presentation is my proposed research design that could expand on the previous research study to help further understand our understanding of ASD's hereditariness. So the study that I just went over looked at the gray matter volumes of mothers of children with ASD, and they did this to avoid confounding factors related to sex. But to get a more complete sense of ASD's effects on parents of children with ASD, I would propose a similar research design, but instead also look at the gray matter volumes in fathers of children with ASD. Because ASD is one of the most heritable neurodevelopmental disorders, by studying both parents of individuals with ASD, we can better understand its different endophenotypes. And furthermore, since genetics play a crucial role in our brain's formation, particularly in the early stages of our development, from cells to synapses to neural circuits, any complications in our genes can lead to complications in our brain's development, which can thus lead to an inability to respond to our environments. So studying fathers of children with ASD will provide more evidence for understanding the broader autism phenotypes of individuals who may either carry the cognitive traits of ASD, but have a more mild version of it. And I propose that by incorporating some genome transcription techniques, it can help uncover potential genetic mutations that may lead to improper performance of neural activity and functional issues in our brain regions. And by pairing genome transcription techniques with VBM analysis, like in the previous study, we can expand on Ann et al.'s research by further investigating the relationship between brain imaging and gene expression in those with ASD and parents of those with ASD, and further close the gap in our understanding of its heredity. So that closes my part of the presentation, and now I'm going to turn it over to my group partner, Deba.
All right. So now we'll be getting into my section of the presentation. So we know autism to be a pretty complex neurodevelopmental condition, but not much is actually known about its neurobiology. The focus usually tends to be on the social, communication, and cognitive difficulties that are associated with the condition. However, another key domain of the autistic experience has been brought to light with recent revisions to the diagnostic criteria, and that is sensory processing. There's been evidence that supports the idea that sensory symptoms manifest themselves pretty early in development. So they contribute a unique variance to diagnostic criteria and drive our need to understand the neurological basis of autism. So a term we're going to become familiar with in our full presentation is sensory integration and how that's impacted in the low-level processing areas of sensory dedicated regions in the brain, causing autistic persons to either be hyper or hyposensitive to sensory input, affecting their perception of the world around them. Worth noting is that this looks different in everyone with ASD, and there's no singular manifestation of the condition. And we can see this evidence even in genetic animal models, which we'll talk about later. So in part 2 of examining sensory perception autism, we'll also look at the ways in which this gap in knowledge is being addressed, and perhaps more importantly, the insights gained from attention to these behaviors and how these may have important implications for developing animal ASD models. And examining how these sensory issues arise and mapping them out into other models, we may be able to more closely see the relation to other behaviors that characterize autism and incorporate more sensory-based interventions into the treatment profile for ASD. I'll also go into further detail about a proposed experiment in which I'd like to gather more detail on the sensory effects of autism, more particularly how the auditory cortex is affected. So the ability to reflect on our own thoughts, our own and others' thoughts and emotions, which we referred to as theory of mind, which Lenny referred to earlier, is a defining characteristic of human cognition. Children with autism spectrum conditions show delays in the development of this capacity, which has consequences for social cognition over the course of a lifespan. And a lot of the time, these alterations in social cognition are accompanied by a very different perceptual experience of the world. And atypical sensory experiences are estimated to occur in as many as 90% of autistic individuals. And they're known to affect every sensory modality, which is taste, touch, hearing, smell, and vision. So autism is often posited as a disorder of the social brain, but now researchers believe that sensory differences represent alterations in domain general mechanisms like attention rather than secondary consequences of reduced social interaction. Sensory traits are, in fact, core phenotypic markers of autism, and sensory symptoms have been clinically documented as early as 6 months of age in infants later diagnosed with autism, which is considerably earlier than children when they reach key developmental milestones in social cognition like joint attention. So in the review paper I read, there was evidence suggesting that the autistic cortex is affected by distinct low-level changes in neural circuitry that is dedicated to perceptual processing, which includes the primary sensory areas. So the idea is that an understanding of the perceptual symptoms in autism could provide insight into signature differences in neural circuitry that might underpin multiple levels of autistic features, and that could help us to explain autistic neurobiology, which I'll get into more later. So children and adults with autism, as well as those with other developmental disabilities, may have a dysfunctional sensory system, which is referred to as sensory integration disorders in ASD. And sometimes one or more of these senses are either over or underreacted to stimulation, and such sensory problems may be the underlying reason for such behaviors like rocking, spinning, and hand flapping, which is explained by the term stimming. All people engage in behaviors to help regulate their sensory systems, but when these behaviors are overt, they become known as stimming. And the amount and type of spinning used can vary a lot from person to person. Most forms of stimming are repetitive or unusual body movement or noises, as I mentioned earlier with the hand flapping or rocking. So getting back to our definition of autism as a sensory integration disorder, sensory integration is an innate neurobiological process, and it refers to the integration and interpretation of sensory stimulation from the environment from the brain. In contrast, sensory integrative dysfunction is a disorder in which sensory input is not integrated or organized appropriately in the brain, and this could cause a degree of problems in development, information processing, and behavior. So sensory integration focuses primarily on 3 basic senses: tactile, vestibular, and proprioceptive. Although these 3 sensory systems are less familiar than vision and hearing, they are just as critical to our basic survival. Basically, they allow us to experience, interpret, and respond to different stimuli in our environment. So the tactile system includes nerves under the skin surface that send information to the brain. This information includes like touch, pain, temperature, and pressure. These play a pretty important role in perceiving the environment as well as protective reactions for survival. A dysfunctional tactile system may lead to a misperception of touch and or pain, and may lead to self-imposed isolation, general irritability, distractibility, and hyperactivity. The vestibular system refers to structures within the inner ear, otherwise known as the semicircular canals, that detect movement and changes in the position of the head. So dysfunction of this sense may lead some children to be hypersensitive to vestibular stimulation and to have fearful reactions to ordinary movement activities, such as like turning the head or so. The proprioceptive system refers to components of muscles, joints, and tendons that provide a person with a subconscious awareness of body position. So some common signs of proprioceptive dysfunction are clumsiness, a tendency to fall, or a lack of awareness of body position and or odd body posturing. This next slide is a summary of the findings regarding the research that was done into the sensory experience of autistic individuals in the review paper that I had read. So what they had found was that individuals with autism have sometimes been described as seeing the trees, but not the forest. This means that they are attuned to the details of the perceptual world at the expense of the global perception they compose. This is also saying that perceptual representation in autism shows a relative bias towards local over global features of a sensory scene. And this detail-focused perceptual style is well captured by two studies of autistic visual behavior, in which the first found that individuals with autism show faster detection of singular details among visual distractors. And the second showed that gaze patterns from individuals with autism during passive viewing of naturalistic complex scenes favor scene regions that rank high in pixel level saliency. And to elaborate, that these would be regions that are salient in terms of contrast, color, orientation, which is the stuff you would pay attention to as compared with object level saliency or semantic level saliency, which you can see down there in those last two boxes on the left, which usually drive gaze biases in neurotypical individuals. And consistent with the psychophysical evidence, which indicates a low processing level origin of the local global perceptual sign autism, neuroimaging and evidence also strongly suggests that autistic sensory traits are indeed low level in origin, and atypical responses and representations in primary sensory cortices have been observed in autism across sensory modalities and during multimodal perception. So in the visual cortex, which you'll see more so on the right, the gross organizational properties of visual areas are typical in terms of the surface area devoted to each visual cortical region, which would be V1, V2, V3, and V4. Retinotopic maps, like we talked about earlier in previous lectures, and the cortical area that is dedicated to each part of the visual field. However, there are distinct differences in neurochemical composition and signaling accuracy of early visual cortex that has been observed in autism. And GABA, which is a neurotransmitter we've talked about, has actually been implicated in visual suppression deficits in autism. And the tight linkage that is usually found between the strength of visual suppression and GABA levels in the visual cortex, including V1, in neurotypical individuals is absent in autism. There's also larger population receptive fields in autism and cortical responses evoked by sensory stimuli like moving dots, auditory tones, and tactile stimuli, and they're less reliable in individuals with autism. Given all this, there's evidence that may support the idea that GABAergic signaling pathway is key to the neurobiology of autism. It's also been found that there are reduced GABA levels in the auditory and somatosensory cortex of autistic individuals, which suggests that reduced inhibition may characterize several cortical regions and perhaps underlie several sensory traits in autism. Yet it's still unclear which part of the GABAergic pathway might be atypical in autism, and we won't necessarily get into that with our presentation, but we're just showing that it has been implicated in the sensory side of autism. So given all this evidence, it's pretty clear that perceptual symptoms may be a great tool for translational research, especially in animals and mapping that on animals. And I'll go into more detail about regarding the theory behind this now. So research at the animal level can help to motivate the design of sensory paradigms in humans. And that can help us to find those alterations in neural circuitry that produce the dysfunctional behavior traits in humans. And this can extend to other domains of autistic traits outside of these sensory systems. A crucial next step would be to test whether genetic models of autism capture human level behavioral differences using identical sensory paradigms. And it's now been suggested that a standardized set of sensory paradigms that demonstrate clear psychophysical differences in humans and that are suitable for translation into animals would also help to enable validation of animal models in autism and further research into the neurobiology of the autistic condition. And also, because of how genetically heterogeneous the autistic population is, one promising contribution of sensory paradigms is that they would allow us to arrange the autistic population into more homogeneous groups of people who have shared underlying alterations in the neurobiology. So we're just seeing the sensory system and the effects it has, and looking at these alterations in the circuitry, we can have that mapped out. We can have a greater understanding of the overall picture of autism and how that extends beyond the sensory side. And there's also good news in that sensory strategies, taking treatment from this approach, we can use this for treatment, and some adjustments in environment can be made. The challenge, however, is that every child or adult with autism has a very different sensory profile, and they'll require their own unique set of supports. In this case, an occupational therapist would probably be the best professional to provide support, as they could apply a sensory-based approach, which would concentrate on correcting or improving the body's abnormal response to external stimuli. And interestingly enough, sensory integration therapy is actually the third most common form of treatment used by parents of children with ASD. So we're seeing there's a growing popularity of this. We're starting to incorporate more of that sensory side when we're looking to treatment of individuals with ASD. And in this type of therapy, children are evaluated to see if they're hypo or hypersensitive into what sensory stimuli. It also addresses the proprioceptive and vestibular senses, as we talked about earlier, which is really nice that we're having this more full picture. We're incorporating all the senses, even those that aren't talked about as often, but that are just as important. Though there's still a need to do some expanded research to have a more accurate picture of the effectiveness of sensory integration therapy. There's still been a growing use of this kind of therapy, which is really good because it's showing the growth in the knowledge, and we're just kind of helping to expand on that and show the research that has been done to allow for this. So right now, we know that in this part, I'm going to be going into the experiment in which I'd like to close this gap, especially in regards to how the sensory side is affected. So right now, researchers know of disruptions in the temporal envelope of sensory processing that have been observed in the domain of auditory processing in autism. Researchers also have some evidence that supports the idea that autistic children show delayed evoked neural responses to auditory tones as compared with typically developing children. I'm really curious to looking at the delays in other areas of auditory processing, like auditory discrimination, which is basically how well you can tell the difference between two sounds. And I'd like to test this hypothesis in an experiment of my own by testing a group of 60 children ranging from the ages of 6 to 18, because this is the population that's most affected by autism, or in which most testing is done. And I would divide them in half by whether they have an ASD diagnosis or not. It's also important for them to have a normal or corrected to normal hearing, because this is an auditory task. And what I'll have them do is I'll have them wear headphones in a quiet room, and they'll be presented with auditory stimuli. They'll hear two different tones, one presented in each ear, and the tones will be presented in close temporal proximity. So we'll hear them pretty close to each other. I'll then ask children to respond with their answer to that question of which tone was louder. And this experiment will help to test their auditory temporal acuity, as well as their auditory discrimination skills, and help to lessen the knowledge gap we have regarding children with ASD's auditory processing skills. Because I would imagine that there would be delays not only in the temporal side, so it'd be harder for them to identify these differences if they were farther apart, and also if the differences in sound were very close together, it'd be a little difficult for them to tell. But that is the experiment I would propose. And with that, we have reached the conclusion of our presentation. I hope you guys learned a lot today about the multifaceted nature of autism. Thank you for listening.